Thank you very much, Mr. President. It's really a delight, and actually when I got the news last year, I couldn't believe it. I mean, British science and microbiology is such a big thing for me, and being recognized at this level is really a great thing. So thank you very much for your kind introduction, and thank you for being here. What I'm going to do, and maybe I need to decrypt my cryptic title a little bit, is to sort of take you through basically what we've done over the last years, is to try to go from what was mentioned, which is, let's say, the cellular microbiology of microbial pathogens, which is Shigella, and basically try to move or translate this concept that we've learned by doing this into the concept of microbiota and homeostasis, and basically back to pathogenesis. So this is a bit of an exercise. My training is in microbiology and medicine, and you know, I like this idea that we are living in this balance between homeostasis, physiology, and pathogenesis. So that's what my title is about. So the first thing is, of course, Prince Hamlet has questions about microbes, and the question is basically what has happened with these microbes, with us. Basically, microbes came on Earth 3 billion years ago, and complex multicellular eukaryotes appeared on Earth about 800 million years ago. So they preceded us by a long period of time. And it's clear that as we progressively emerge as what we are today, we had to cope with microbes. The question is, did we just ignore them, or did we decide in a way to make a piece of path with them and try to develop some symbiotic interaction? And of course, this is what it is. This coevolution has led, with multiple, I guess, tries natures, like, as Darwin said, to recycle good solutions, set up a very robust interaction with this microbiota that is present in humans and, of course, in mammals and mostly all animal species. So as Theodosius Dobzhansky said, I mean, nothing in biology makes sense except in the light of evolution. And we are really trying all the time to adapt our questions and hypotheses to this concept that everything that we see in this case is the result of coevolution. So just to make a long story short, and this is summarizing lots of work before and after the metagenomic era, the gut microbiota, I'm going to focus, of course, on the gut microbiota, is a huge assemblage of microorganisms. As you know, there are two major families, the firmicutes, anaerobic gram-positive, the Bacteroidetes, anaerobic gram-negative. But beside this, and probably in less number, but doesn't necessarily mean that they are not important, there are also Proteobacteriaceae, Actinobacteriaceae, and Archaebacteria, and they all sit there for some purpose. And we would really like to see how all these microorganisms at some point crosstalk. If you wish to do this, which is a very ambitious endeavor, probably beyond my capacity in times that's left in my career, I like to focus on niches, model systems, and that is what my talk will be about. I'm not going to get into all these numbers, 10 times more prokaryotes than eukaryotic cells in the body, 300 now times more prokaryotic genes than human genes. These tells you a bit about the importance of this symbiotic interaction, but it doesn't go beyond this. So far, this sort of metagenomic science has been extremely exciting. It has got us through descriptive phase, through correlative phase between some diseases and changes in the microbiota composition, what's usually caused dysbiosis. But I think that the time has come, as me and others used to say, to experimentomics, which is to really address the questions in experimental terms. And that is the idea of basically switching from pathogenesis, cellular microbiology of pathogen, in crosstalks with eukaryotic cells, towards a cellular microbiology of symbiosis or mutualism, if you like. Problem is that it's a little more complicated because a pathogen does things that are very visible, like it kills a cell, so it binds to a cell, so it invades a cell. A symbiont does things that are a lot more subtle, more metabolically based, and the readouts that you can establish, if you wish, for instance, to screen a library of mutants, are a lot more complicated, and sometimes, it's not a black or white thing, as we often see with pathogens. We are sitting in this gray zone, which is complex to interpret. So just to make all this sort of wrapped up in a single slide, I usually show the microbiota to the students as sort of the content of a gland, and the epithelium of the gland being, of course, the intestinal epithelium. So this microbiota is putting a lot of pressure on this epithelium to produce mediators, cytokines, chemokines, neuromediators, and all this is going to regulate a lot the local mucosal physiology as well as, the systemic physiology. Not only these molecules are produced, but also some bacterial components like peptidoglycan, endotoxin are passing through, translocating the epithelium, and they are found actually in the serum. They participate probably as hormones to this global process of symbiotic interactions. And as time goes, a lot of expression of these symbiotic interactions are shown, like, of course, the maturation and maintenance of the immune system, some elements of the metabolism, nutrition, digestion of complex sugar, production of short-chain fatty acids, and so on and so forth. Also, bone metabolism, the brain is in relationship with the microbiota through the maturation of the blood-brain barrier, through elements of late maturation that basically possibly dictates the behavior, and this is clear in the work of Sven Petersen, for instance, in the mouse. And we have shown recently that the microbiota is also important to help repair of epithelial damage under some cytotoxic lesions, and I will come back to this. So then it raises a question, which is, this coevolution has put some sort of a conundrum there, because on the one hand, we have to sort of live in peace with these symbiotic microbes. This is called tolerance. This has to do with the establishment and maintenance of this state of physiological inflammation, as Mechnikov called it, which is the state of preparedness of the immune system to quickly respond. So this is part of the mutualistic interaction that we are talking about. And if there is a disruption here, of course, there might be some side effects that occur, like inflammatory bowel diseases or possibly colon cancer, for instance. So this symbiosis is very robust, but there are evidence of weaknesses sometimes, and these are elements which are very important to take into account. On the other side, we have the pathogens. They are engaging the epithelium. They are causing pathological inflammation. That's part of the innate immune response, as well as the adaptive immune response to get us protected at some stage. But the question is, how is the host, through its immune system, able to discriminate between, let's say, a good bacterium and a bad bacterium? Because after all, they are bacterium. They have lipopolysaccharide for the gram-negative. They have peptidoglycan. They have lipoproteins, flagellin, whatever you like. And the sensors, as we know them, they recognize these PAMPs or MAMPs. They are not by themselves able to say this one is a bad guy and this one is a good guy. So I've been really teased by this question for many years. And of course, I guess it's obvious for many of us, the way the discrimination is carried out by the immune system, the way the host can distinguish a pathogen within seconds compared to a commensal is the presence of these danger signals. And this was mentioned in the introduction. Over the years, we've really used Shigella as a way to ask these questions about how a pathogen is perceived by the host and responds accordingly. So this is a slide that sort of summarizes 30 years, maybe 35 years of work. It's a bit of a shame, but that's how it is. Shigella goes through the help of this type 3 secretion system. I'm very short here. through the intestinal epithelium. It causes the killing of these macrophages here, which allows the bacteria to survive in the subepithelial tissue, and then it goes through this round of cell biology numbers like entry through actin polymerization, lysis of the phagocytic vacuole, cell-to-cell spread through actin polymerization again. So all this made our bread and butter, as Stanley Felker uses to say, in the 80s and 90s. But soon we started to realize that this was a nice cell biology exercise, but it was missing maybe the real matter, which was inflammation and tissue destruction. So we got there in the mid-90s, starting to figure out that the epithelial cells were actually reprogrammed by invasive Shigella to become pro-inflammatory, that the immune system was also involved. Macrophages, again, were killed by this process of pro-inflammatory apoptosis, as we called it at the beginning. So all this was starting to make some sense in comparison, of course, to what was seen in experimental models. So this notion of danger signaling or sensing of pathogenic signals, I think we went through this route thanks to three basic observations, which I will very briefly mention. The first one goes back to 1991, 1992, when Artur Zischlinski was a postdoc in the lab. He had worked on apoptosis at the Rockefeller, and he said, what you see is this quick killing of macrophages, it is apoptosis. So I said, apopto what? And he actually came to the lab, and in a few weeks or months, showed that actually invasive Shigella was killing macrophages by this apoptotic process. We subsequently showed that it was one of the type 3 associated molecules, IPA-B, which was causing this. And this associated with caspase-1 and activated caspase-1, which itself induced the cleavage of pro-IL-1 beta and pro-IL-18 into the mature forms, which were secreted. And this, within minutes, was causing massive inflammatory response. And if we treated the animals with anti-IL-1 beta, we basically not only blocked inflammation, but we also blocked invasion. So it induced the idea that the bacterium at the beginning of the process needed to raise some inflammation to disturb the epithelial barrier and invade. So that was the first signal of a danger signal. And the second was, as was mentioned in the beginning of the 2000s, Dana Philpott, a postdoc from Canada, came to the lab, and we started to work on why is it that an invasive Shigella doesn't induce or very slowly induces epithelial cells to start producing pro-inflammatory cytokines like IL-8, and an invasive microbe which penetrates into the cells and escapes into the cytoplasm actually induces massive expression within minutes of interleukin-8. And through this, and I'm making a long story short here, we, with Stéphane Girardin and Dana, came up to recognition that the NOD molecules were actually cytosolic sensors which were sensing the presence of intracellular bacteria, which is by definition a signal of danger. It's an abnormal situation. And we could, of course, demonstrate at the time that peptidoglycan was the bacterial molecules, the agonist of the NOD molecules. Signal, danger signal 2. Danger signal 3 is more recent. This is the work of a postdoc from Austria, Andrea Puar. What we showed, again, to make a long story short, is that as Shigella, the invasive form, is engaging intestinal epithelial cells, it's causing these cells to release massive amounts of ATP from the cytoplasm through these heme channels, which are hexamers of connexins. And ATP is very good inside the cells because this is the basic source of energy. ATP is very bad in the extracellular milieu outside the cells because it's a very potent pro-inflammatory molecule. So you don't want to have ATP in the extracellular milieu. And this is what Shigella is causing at the very beginning, which is to induce the massive release of ATP. This is sensed by cholinergic receptors, for instance, and causes a cascade, a sort of vicious circle of events which lead to the accumulation of these ATP molecules in the tissue, and we could show that in vitro as well as in vivo, and it's actually the case for Salmonella as well. As soon as cell bacterial pathogens start, teasing the cells and playing with the membrane and inducing some calcium fluxes, they lead to this release of ATP. So this is sort of danger signal number 3, and there are others, but I just wanted to show you this as a sort of an example. Of course, at some stage, what we also learned from Shigella is that it's very dangerous to play with matches. So it's igniting this massive inflammation, but that's going to kill the microorganism through recruitment of phagocytic cells, overexpression of antimicrobial peptides, and so on. And thanks to this, we could basically demonstrate that there are effector molecules which are expressed by Shigella, which are secreted through the Type Three secretion system, which shut off the expression of most of these pro-inflammatory systems and basically manage a situation with time in which the bacterium initially induces inflammation and then at a certain moment starts to basically attenuate this inflammatory response. And this has an impact on the adaptive immunity as well, and probably explains why Shigella is such an immunosuppressive microorganism and basically leads to these weird immune responses like the T-cell response, for instance, is very poor, which is not a good thing to control an intracellular pathogen. So that was sort of how we went through this in very short terms. But then we started to think about, at the end of the day, where is the bacterium really invading? In the patients, kids that get shigellosis, the invasion process occurs in the colon. So we started to sort of move our systems towards the colon, and this is a piece of human colon. That we see from the top. So the dark holes here corresponds to the entrance of the crypt. Everything else is the autofluorescence of tissues. They've been infected in this ex vivo model with fluorescent Shigella. And what you will see on this animation, if it decides to start, yes, is, okay, I guess there is no need for comments. You see that the bacteria are essentially present in the crypt. And they can go quite deep in these crypts, actually, down to the bottom, where, again, the regenerative apparatus of the gut epithelium is located. So that was an interesting observation, and we sort of furthered this with Elena Arena, a postdoc from the US, and Jean-Yves Thiefnes on the campus, on the Imagopol on the campus. We have a nice now model of guinea pig that reproduces the symptoms and pathology of Shigella infection. And by educating the computer after reading the localization of the fluorescent microbes in the course of invading the colon and looking basically at the entire surface of mucosa that was explored by the microscope, we could come up with a sort of coordinates of all the microorganisms with relation to the epithelial surface. And as shown on the next slide, basically we could follow with time the trip of a non-invasive and of an invasive microorganisms and basically show that as the non-invasive microorganisms disappears from these crypts, this is all averaging, hundreds and hundreds of different crypts that have been photographed. You could see that after 8 hours, the bacteria were actually, on the average, penetrating very deep down to the bottom of the crypts, as well as sort of starting to invade the tissue more at the level of what we call the mouth of the crypt. So this is the kind of new tools that we need if we want to bring this field of cellular microbiology towards what we now call tissue microbiology, even if it doesn't sound as well as cellular microbiology. So we were getting very much into the elements of, looking at the crypt, and the crypt turned out to be an interesting area to study because it's one of the rare situations in which probably stem cells are associated or in contact with microorganisms or very close contact with microbial products. So the crypt is, has shown here on the small intestine and in the colon. It has this regenerative structure with the stem cells and the Paneth cells, which are nurturing them and providing some growth factors like orspondin or Wnt ligands. And then you have the proliferative compartment where cells cycle very quickly, and then suddenly the cell cycling stops and the cells start to differentiate into the lineage that are characteristic of the gut. So we started to wonder whether beyond this massive amount of microbe in the lumen, there might be some particular microorganisms living in these crypts. And to make a long story short, yes, there are in the cecum and in the colon. There are very, very few in the small intestine. And we started to try to make sense out of this and whether we had been able to identify a symbiosis inside the symbiosis, which would be what I mentioned at the beginning. What is the function of these bacteria that are more as a minority, but still might develop in particular niches, some very particular symbiotic interactions. So we started with Thierry Pedron in the lab and Céline Mullet in the mouse for the moment. And we looked at the presence of microbes by Wharton starry staining. And yes, there were in the cecum at the bottom of the crypts, in the proximal colon and quite far in the distal colon sometimes. And because these could be dead microbes, we did fish analysis with universal primers, and they were not dead, they were there living. So there was clusters of microorganisms at the bottom of the crypt, especially in the cecum and in the colon. So we started to try to identify these microorganisms. We went, of course, by molecular study. So what Thierry did was to establish a sort of pipeline, as we say now, a platform to be fashionable, where they microdissected the content of the crypt with a laser capture microdissection system, amplified, sequenced, and came up by a 16S DNA analysis with a certain number of OTUs, which were dominated by completely unexpected phyla, Acinetobacter and Dalftia comamonas. Why unexpected? Because we are in the gut, that is supposed to be totally anaerobic. These are strictly aerobic, non-fermentative microorganisms. So the first comment of the reviewer was, this is contamination. So, okay. We decided to go for the hard work, which is to do real microbiology. So we washed the surface of the epithelium with a bit of bleach and, managed to contain or maintain, a piece of tissue that would correspond to the crypt from which we could isolate these microorganisms. And believe it or not, we found them. And they grew. We got this Acinetobacter. It's a gamma proteobacterium. Again, it's strictly aerobic, non-fermentative, highly biodegradative. We got Dalftia acidovorans, which is a beta proteobacterium, and a few others, but these were really the two major, species or genus that we identified here. So now we had these microorganisms, and we needed to do some experimentaomics. So we started by taking germ-free mice, and we colonized them, or gavaged them with Acinetobacter, isolated from the previous ones. And what you see that within 2 weeks, the Acinetobacter actually localizes here. So it has a tropism for the crypt. And if we co-housed germ-free mice with conventional mice, basically within 3 or 4 weeks, we also observed the same thing. So there was something here that said that some microbes were adapted to the crypt environment. And then I met Amy Pedersen at a meeting in Edinburgh in Scotland. She's interested in wild animals. I mean, rodents living in the wild for interesting reasons, especially if their immune system actually operates differently from those that are grown in the 5-star hotels in our animal facilities. And we also still had the possibility that there was a bias the way these mice are, housed and fed and so on and so forth. And we were very surprised and actually positively surprised to see that these Acinetobacter, actually we could find them in great numbers associated with the crypt. This is a fish analysis with a universal probe. So you see all the bacteria here and of course there. And here it's a specific Acinetobacter probe. So they go there and they live in the crypt. And I've just added this very nice picture here because I think it says a lot about the fact that these Acinetobacter are actually living as a little cluster in the bottom of the crypts. And you would like this of course, to represent some sort of a symbiotic interactions, and this is what we are doing at the moment. So why oxygen or aerobic microbes? With Christang and Benoît Martin, several years ago, we've shown that there was oxygen at the surface of the intestinal epithelium. I'm not going to get into the detail, low concentration on a sort of layer that probably corresponds to oxygen molecules that have diffused through the epithelium, which are trapped by the mucus. So this might turn out to be an interesting discriminating zone between aerobic microbes and anaerobic microbes. So that's for oxygen. Aerobic microbes, non-fermentative, we've shown that some products of fermentation like propionate or acetate, for instance, they do block cell cycle. So this is not a good thing probably for the regeneration apparatus. And these microbes are highly biodegradative. They are microbes from the environment. So it's a bit like in the field of plants. Maybe we have actually harnessed some microbes from the environment to establish them in the crypt and basically in this coevolutionary process maintain homeostasis of the crypt. And you are very much looking at this, especially the role of these microbes in protecting the stem cells against the genotoxic stress by xenobiotics from the food, from the environment in general. So this is where we are going actually in this field. The last part of my talk will be about the more, again, experimentomic aspects of things. And this is a work by Julia Negro, a very, very talented postdoc, and now she has a staff position at Institut Pasteur. We started to sort of ask questions in vitro or ex vivo about how microbial products or microbes can actually interact with this, strategic element of epithelial regeneration. So we borrowed from Hans Clevers in the Netherlands this technology of organoids. Some of these mice, they are transgenic mice. They express GFP under the control of the LGF5 promoter, which is GF5 is a R-spondin receptor, so it's specific to the stem cells. So we sort of started to gather tools to look at these interactions, again, in this sort of cellular microbiology mood that I was mentioning before. So the first experiment that Julia did actually worked immediately, and I was, when experiments work the first time, you start getting concerned, but it could be reproduced. And what she showed is that if you expose these organoids, you take the crypt from the mouse that has been just sacrificed, you transfer them into this matrigel substrate. You associate a lot of growth factors like R-spondin, for instance. And at the same time, she added microbial PAMs or MAMs like peptidoglycan, muramyl dipeptide, muramyl tetrapeptide, because we are in the mouse background, lipopolysaccharide, flagellin, PAM3, which is a TOL2 agonist, and the controls. And we could see that there were more organoids formed. The yield of organoids was a lot higher when peptidoglycan, or at least muramyl dipeptide, was added to these organoids. And this was a sign that there was something going on as we analyzed it, not so much in the sort of proliferation that leads to the formation of the organoid, but more in the survival, the protection of the stem cells. So we went further, or Julia went further. And of course, since we are talking about muramyl dipeptide, we are talking about NOD2, which I mentioned before. And NOD2 is a pro-inflammatory molecule. It's also a molecule that is somewhat related to establishment of autophagy. So there are lots of things now that we need to look at with regard to how this MDP is actually inducing this protective mechanism through NOD2. But we still had to demonstrate that NOD2 was expressed by the stem cells themselves. So this was done by developing techniques to purify these adult stem cells from the mouse intestine, from the mouse crypts. We also have access to NOD2 knockout mice, which interestingly enough have GFP in the first exon of NOD2, so it's phenotypically NOD2 knockout, but you can see the cells that express NOD2 at high level because they have GFP under the control of the promoter, and sure enough, it was essentially these small cells squeezed between Paneth cells which are the stem cells which were concerned by the expression or the high expression of this NOD2 molecule, and we could actually demonstrate that the level of transcription was a lot higher, and that all this protective mechanism was lost when we worked with NOD2 knockout mice, showing that there was a loop of MDP, NOD2 cytoprotection. But people in the lab were not so happy during the lab meetings because they said, well, it's a factor of 5, protection. What does it mean? It's not enough. So we started to think that there might be another angle to look at, and this might be related to some thinking. And there were a discussion yesterday after a talk about, lesions to the gut and tissue repair, and that if this doesn't operate properly, there might be, a loss of the epithelial barrier functions and inflammation accordingly. So what we did was to basically treat the mice 3 days before the sacrifice with doxorubicin, which is a DNA intercalating agent, which causes massive DNA break and oxidative stress. And this is a killer for the cells when it goes to that level. And doxorubicin is an anti-cancer drug by definition. So doxorubicin actually, interestingly, is autofluorescent. It fluoresces in the red band, and it actually associates also with the stem cells. So it looks as if the stem cells have some capacity to incorporate at higher level this kind of drugs, and this is part of the work that we are doing at the moment on efflux pumps and so on. So we haven't yet really characterized the physiology of these cells with regard to some pharmacological elements. And sure enough, when we did this, we could show, if we took these organoids again from the Crypts, that the level of protection now was a lot higher, like 15, 20, even more if we change the experimental conditions. And this actually related to a much higher capacity of the animal to regenerate its epithelium after these lesions. These are in vivo experiments. We look at, for instance, EDU staining that reflects the level of cycling in the transit amplifying compartments, and it was a lot higher in the wild-type animals compared to the NOD2 knockout animals after this treatment with doxorubicin. So this was sort of the first example in a way that bacterial products, MDP in this case, is cross-talking with stem cells and actually playing a role of cytoprotection upon some damage caused by doxorubicin, which was here taken as a model system, but it could be an infection, it could be a xenobiotic, it could be a lot of other things. And this is actually opening a new angle with regard to Crohn's disease, for instance. It's known that the familial form of Crohn's disease often have a mutation in the NOD2 gene. And so far, it wasn't so obvious what happened, because in a way, NOD2 is a pro-inflammatory system. So how is it that with a knockout, a loss of function mutation, you get more inflammation? And maybe the idea is that we are playing here not so much with the subtle regulation of inflammation, but more with the regulation of epithelial regeneration and repair. It's possible that the wild-type individuals that don't have the NOD2 mutation, whenever they get a stress on their crypts by infection or toxic products, for instance, they can repair properly because they can feed on NOD2 and muramyl dipeptide from the microbiota to proceed at a level that will regenerate the epithelium properly within a few hours or days. Whereas in the case of the NOD2 mutation, it's not unlikely that the people cannot regenerate their epithelium because cells are dead, they are not protected. And the consequence, of course, is a disruption of the epithelial barrier, and the microbiota now can put some pressure on the immune system and cause inflammation. So we are more and more in this trip, I must say, to figure out the pathogenesis, and we are constructing new transgenic mice to address this. So this is the last, I would say, last part of my talk, just to show you that cellular microbiology is on its way. So we had Tomoaki Naito, Tomo, in the lab for 2 years, who started to look at the interaction of these microbes, we call it the Crypt-specific core microbiota, CSCM, Acinetobacter, and others on the regenerative apparatus. And I will just summarize his work in 2 slides. Actually, we have purified to high purity grade these PAMPs from these microorganisms, especially endotoxin, epithelal, with Antonio Molinao at the University of Naples. He's a master in this. And then we have treated organoids with these elements. And to make a long story short, and I summarize in 2 slides, the endotoxin of these microorganisms, the lipopolysaccharide, is actually causing a decrease in the number of organoids, and more specifically a disappearance of these external modules here that form on the organoids, which are linked to the presence of the stem cells. And to make a long story short, I'm not going to show all the experimental details. Endotoxin is actually, at least the one from these microorganisms, is cytotoxic to a certain extent to the stem cells, and this is slowing down the regeneration apparatus. So now we have to start thinking in terms of balance. I mean, it's like vectorial geometry. You have vectors go in one direction, others that go in another direction. It's the resultant of the vectors which is going to represent the homeostasis of the crypt, and we're extremely interested in this, even though it's a kind of a complicated business. But also another important observation that he made is that endotoxin not only is sort of down-regulating intestinal proliferation, but it is also leading to changes in differentiation of the cells, especially in leading to higher expression of goblet cells. And we are now looking at the molecular basis of this. So, as summarized here, it's kind of, as I said at the beginning, or I tried to say at the beginning, trying to switch from cellular micro of pathogens to cellular micro of symbionts. It has limitations. It is more complicated, but I think with these experimental tools that have been developed over the last few years now, we may progress and ask questions again about the pathophysiology of this, what's so-called postmodern diseases like inflammatory bowel diseases, which are clearly associated with dysbiotic elements. So I will stop here by essentially mentioning my institutions. These were already on the first slide: Institut Pasteur, INSERM, and the Collège de France. These are the people who participated in this work. The red ones are present in the lab. The blue ones have left, and they're continuing their work somewhere. These are our collaborators on the campus, and several collaborations in France and outside. And again, I mean, they are listed here. And Chris Tang, for instance, we've been doing lots of things on Shigella genetics over the years, very productive and friendly interactions, but there are also others which are mentioned. So thank you very much again. I'm delighted to be here, as I said, and this is 35 years of a career in 35 slides. That's the best I could do. Thank you.